Welcome everybody. This is the second in our series, and we'll have one more before springtime. But today we're going to talk about the seed. Springtime, so everybody's getting a little bit anxious. And just put this in here. We've got that seed sitting in the bin, and everybody's excited, ready to roll. And we're going to just take a look at the whole perspective around seed. And I want to make some general comments about seeding. Basically, you know, for all the years that I've been involved in this, about 15 since I was with Agritrend, still seeding has been one of the biggest problem areas that we faced every spring. You know, too deep, too shallow, you know, clumping on the air seeders at the ends wherever it is, poor soil seed contact, variable emergence. And those are all sort of negative factors relative to getting good seed germination and crop growth. And I'm going to be addressing some of those throughout in terms of how the plant sees that. But, you know, those are kinds of things that we can't deal with all the time. Farmers are in a hurry. And it kind of explains why the corn guys have done a good job and why some of the folks that are using planters for canola now and planting at a pound or pound and a quarter an acre can get really good stands because their seed placement is basically perfect, so they get very little mortality. A lot of folks plant at 5 pounds an acre or 4 pounds an acre with canola, expecting 50% mortality. Well, that happens a lot. And one of my contentions is that cereals, the mortality rate of seedlings and seedling death as well as seed death is much higher than the 5–7% that people estimate. So it influences their final plant density, and most people aren't really taking that into consideration. One point I want to make again before we get into this, and I'm not going to talk about seeding after this, is make sure you get packing, because as you put in that seed, and if you don't cover it with any kind of packing or soil material, any water that moves by diffusion from deeper down into that dry area just goes right straight through into the atmosphere, and you can get tremendously variable rate germination because of a variance in terms of the amount of moisture and humidity that the plant can take into imbibe the seed, or the seed can imbibe the water. And that's what actually starts the process of germination and takes it through to fruition. So let's get going. So some general things. The plants we are growing are annual plants. They flower once and they die. And throughout the life of this plant, it's all heading towards producing seeds. So there's progressive senescence until death. And then as the plant dies, it mobilizes and transfers its nutrient to the seed. And then in the springtime, the reverse process actually happens, where the seed then germinates and transfers the nutrients to the new growing shoot. So basically, the growth of the seed, or the nutrition level of the seed, is dependent on the previous year mother plant's ability to store the proper amount and the total amount of seed necessary to get that seed up and going. And the reality is, we have tremendous variability in terms of the nutrient content in that seed across the landscape, and from variety to variety, for that matter, based on the ability of the plant to secure nutrients and then move them. We're going to talk about all those, and the beginning part of the webinar is going to have a little more detail on the process, and then we're going to move to straight photographs of how the whole thing takes place right from the beginning to the end. So what we're going to do is we're going to start off at the back end of this. We're going to start off here at grain filling and what happens there. And senescence in the mother plants is affected by daylight, temperature, genetic and hormones. Plants reclaim resources of N and P and microbes. And for the plant to do that, it has to kill cells, basically. So the chloroplasts are the most dramatically affected, and they're cannibalized. The catabolize is the same as cannibalize. And the plant kills them, and the two nutrients that are moved the most are proteins. And that's a lot of N and P in terms of genetic material and proteins. There's a loss of color because we're taking out all that green from the chlorophyll and mobilizing it till death. And the fact of the matter is, the plant is programmed to die. It's called programmed cell death, if you want to take a look in the literature on that. Let me give you an example. the first thing that is killed in the plant is actually the xylem, because the xylem is produced, then the plant kills it so that the structure is good and solid to take the pressures that are involved in moving nutrients and water up the xylem. Then the plant kills something called a tapetum, which surrounds the pollen grain. It kills that so that it can release the pollen grain. So the plant is perpetually producing new cells and killing off some of its old cells depending on its stage of growth. So this is not unusual, and this is all run genetically. So here we see soybeans, and you can see that as the chlorophyll goes, the plants turn brown, the seed pods, in fact, become brown and mature. So it's taking all that nutrient as best it can, as well as what is coming through the roots. As long as the plant can get nutrients and water through the roots and primarily moved by water, it's going to still continue to pick up some. But when it gets really late, then the plant actually starts moving nutrients out of the roots, and the root sequestering of nutrients and water drops off, and it moves everything basically from the plant into the seed. And that's what a monocarpic plant does. And then eventually we get that. So what happened on the road to senescence is really key to what's in the seed. So here's a wheat plant. You can see it's pretty well dead. And there's a primary head and a tiller. I mean, you know, they're not very great, but that's okay. And that's the end product. So we'll go right to the beginning. We'll go right to the beginning of where this seed develops. And here we have inside a floret of a wheat floret. This is the embryo here. This is the anther, and this is the stigma. So the anther produces the pollen, puts it onto the stigma. It grows a pollen tube down here, fertilizes it, and boom, we get a seed. And here we have the pollen on the stigma, and it's going to grow down and fertilize this particular seed, and that's the beginning of the seed. As the seed grows, and you can see here's a spikelet with 4 seeds in it. And you can see that there's actually a difference in the size of the seeds, and this is the water ripe stage, about anywhere from a week. Probably this one is 5-6 days from pollination, whereas this one, which is the biggest one, is maybe 10 days from pollination. Now you can see that when this plant is going to be finished, there's going to be a variance in terms of the size of the seeds, and there'll be a variance in the terms of the nutrient inside the seed, because this seed is going to be bigger and it has access to more nutrient than this seed, than this seed, than this seed. There's a sequence in which it's pollinated, and I'll show you exactly what that is. Now, for this seed to become really full of endosperm and protein, it has to, after pollination, gather a bunch of nuclear material and cell building material into the seed, and then differentiate it, and it produces what are called amyloplasts to store its starch, because wheat is a huge store of nutrients in its endosperm. There's a germ, an endosperm, and then the seed coat, and that's all. And I'll show you all that inside, but I want to go way back to when it all started. If you don't have moisture after pollination or nutrients that are adequate, things like phosphorus, nitrogen, to build the seed, the number of amyloplasts can't reach maximum potential, and as a result, the seed can't reach its maximum potential to store nutrients. So water after pollination is critical in order to move nutrients into the plant. And I think when we start studying how plants grow relative to soil moisture using some of that stuff that South Country is now selling, we'll get a better idea on how late moisture influences the quality of the seed and the amount of the seed that is produced, the actual volume and yields. I make one other point. I think the time will come where we'll be taking nutrient density from these seeds before we seed it, because that's going to influence just how good your germination is going to be. And it just struck me the other night that we have little control now in terms of the quality from a nutrient perspective in our seed. There's no analysis done on our seed. You get a bag of canola, you don't even know for sure where it came from. I don't know how lentils and peas are sold, whether farmers can regrow those. They probably can. But when wheat, you can. If I was a wheat farmer and I was taking my own seed, I'd look for these 2 kernels. This one, whoops. this one, and this one, and maybe this one. I wouldn't want this one, and I wouldn't want anything from the tiller. So I'd be cleaning real hard, taking that seed and seeding it. And the year before, in the year that I was going to take my seed field, I'd select which field I was going to use as my seed source, probably my best field, and then I'd treat it like a prince. So let's take a look at what happens. So we get the embryo starting to form after a little while, and then we move into this stage, ripening, the early dough, soft dough, hard dough, and full ripe. So we're starting at BBCH stage 8 at the downward end of senescence, and I'll do 8 and 9. And then I'll go to germination, which is stage 1, and stage 2 to the seedling. So we'll cover 4 stages in the growth of wheat plant, and I'll show you soybeans, and I'll show you other things, but I'll use wheat as my primary model. And then the next webinar will cover the remaining parts from stage 2 to stage 8, which takes us through massive growth and through pollination, and then we'll be done the whole cycle in 2 webinars. So here we go. You can see here are the seeds, right from the water ripe right through to mature. And you can see that the embryo is starting while the seed is actually green. It's quite well developed. You can see here is the coleoptile starting here, and we can't see the shoot that well. So this is the milk stage. Here's the hard dough stage, or here's the hard dough stage. This is the soft dough stage, and boom, we get the hard dough stage, and we're done with ripening. So we go to senescence. Overripe, the grain is very hard. Grain loosens in the daytime. Plant dead and collapsing, and harvest the product. So here's what it is. You see, here's that spikelet again that we're looking at. The glooms and the they're not glooms. There's only 2 glooms on the plant. This is the pella and the lemma here, but it loosens off so that you can run that combine through and knock those seeds out. So it's basically mature. Now I want to point out those 2 wheat heads I showed you. What I did is I took out all the seeds and I lined them up. Here's your tiller. You can see why it comes a point that I don't like to see more than about a primary head and 1 tiller, 2 at the most, because that last one is going to produce you nothing and pull a lot of nutrient that should be going into the other 2. So if you're looking at optimal plant densities in wheat, I'm typically shooting for between 30 and 35 live plants at the end of the day. And I won't get into calculations, but I'm relatively convinced in some of the work that guys in southern Alberta have done, Matt Gosling and so on, are showing that in wheat, even down there, we're getting anywhere from 20 to 30% seedling mortality in wheat. But take a look at the primary head, and I want you to look at this. Wheat starts pollinating in the middle of the head, and look at this. Middle of this head, all those spikelets have 4 kernels. As we move away from there, we get 3. We got another 4 there, so that was good, and then into 2s. And as we move to the top of the head, or the spike, we get the same thing, 3s and 3s, and you can see that almost always when you got 2s, the 3rd one is smaller. When you got 4s, the 4th one is smaller. Pretty good principle. So if we want to improve our seed yield, we can get 3s on here, and we can get more 4s on there by providing nutrient for that plant. We got a bigger yield. With wheat, you need seeds and you need heads. It's not like canola where it fills the space. So these are the kernels we want, and we want these 2. These 2 seeds, in all these cases, will almost be exactly equal in terms of nutrient density. And then they go downhill from there. So go at it. Go and get them. Clean heavy, put it on the gravity machine and get those seeds. And here's just another shot of them. So they're mature. Mature seeds ready to go in the bin. So big seeds, more seminal roots or bigger seminal roots. There's not more. There's only 5 in wheat. Faster growth, more fertile tillers, and higher yields, especially under drought stress. So here we are. We got it. The seed is prepared. And I'll just make a few comments about that seed. You know, when the seed is prepared, the plant puts nutrients into it. And it puts in a lot of messenger RNA that fixes the DNA that is broken down during storage. And we're going to go and take a look at what the seed has put in there. What happened on the road to senescence? Well, nutrients were salvaged. We get proteins, lipids, and nucleic acids that are broken down and stored. Major targets are N and P. Nitrogen and phosphorus for building the DNA, P for ATP, nitrogen for all the amino acids that are going to be rebuilt to make this new plant. Just like when I do my stuff on stress, the plant roots are driven by N and P levels in the soil because those two build the cells. If you don't build the cells, you don't have to worry about making them bigger and filling them and that kind of thing in storage. Hydrolytic enzymes are upregulated during senescence to help that break down. N is transmitted as amide, and this is part of my educational part. I'll show you how the amides are transferred because that is really key in both recycling nutrients from the plant into the seed and taking nutrients from the soil and moving them into the seed. It's the same process, only reversed. Largest P contribution from RNA in the ribosomes and DNA. And the largest P contribution is because there's a lot of messenger RNA. In other words, messenger RNA is the code that is going to be used to make the new proteins when the seedling germinates, to make the new shoot. It is the messenger RNA that carries the code. The actual genes carry the code, but the messenger RNA is used to transfer that code to what are called the ribosomes to make the protein. So that's what the plant puts a lot of into the seed. The phosphorus is redistributed to the seed coat and the endosperm. And I'll talk about that specifically because there's a problem there. Other components are broken down and senescent associated genes are upregulated. So here's the cell. Here are the chloroplasts. Those are broken down big time. All of these other organelles are broken down eventually. The vacuole is broken down and cleaned out, and the plant moves that stuff out. In the chloroplast, same thing. All the proteins and so on here in the thylakoid where photosynthesis takes place is broken down. The biggest source of proteins is from this critter, rubisco. Rubisco is the largest amount of protein produced on the earth every day. Every plant on earth uses it to take in CO2 and process it to make sugar. So when the plant is extracting it, it has to kill off the chloroplast to extract all of this protein and some of the starches and move it out. So when it does that, it takes those proteins and it breaks them down into amino acids into ammonia, and then glutamine synthetase and asparagine. And that's exactly the same way the plant handles ammonia coming up from the roots, where it takes nitrate first and then converts it to ammonia, and then it turns it into glutamine and asparagine. So the process is the same all the time. And just for your curiosity, this glutamine synthetase is the actual enzyme that glufosinate inhibits to kill weeds. Because without that, the plant cannot process nitrogen. So anyhow, that's part of the educational part of it. So does the plant die from the top down or the bottom up? Well, let me give you an example here, and I don't know the answer. Elston and I argue about this all the time. But take a look at this little graph down here, this table. What they have is a stay green gene, which is a transgenic gene, which keeps the wheat plant green. So the concept was that would allow the plant to actually move more nutrients into the head and produce more. In fact, what happened, if you take a look at the micronutrients, and this was related to the human zinc project that Dr. Chakmak and a bunch of other folks worldwide are looking at, because zinc is a severe problem globally, and looking at biofortification as a way to get more zinc in. In this particular case, grain protein content in the non-transgenic was a lot higher, but look at the micronutrients that were also higher, a lot higher. So the stay green gene did not perform. In fact, because the plant did not senesce, it didn't have time to move the nutrients from the roots, from all the leaves into the head, and in fact, the program failed. So our plants need to go through this process because they evolved that way over the last several hundred million years. It also takes all the starches and it plugs it into the endosperm and mixes it with the protein. And I put this in for folks because this is a health issue in terms of gluten of late, and diet control. There are two forms of storage. There's the amylose and the amylopectins. And you can see here that amylose is a single branch chain. So for enzymes to work on it, they hook in there and break it up slowly. Whereas amylopectins have multi-sites for amylase to work on it, and you get a sudden release of sugars. So, you know, that's why people will say with bread, it's like eating a couple tablespoons of sugar, because 70% of wheat is amylopectin, so it's broken down very, very quickly, and the glycine index is extremely high. So a lot of work is going on to produce waxy kinds of grains with more amylose, so that the amylopectin isn't broken down nearly as quickly. But in the plant, that is what gives you the sugars to build a new seedling. And there's a whole bunch of proteins that are encoded in terms of senescence, just like glutamine synthetase, for example, is upregulated. I just put there's a lot of proteins, and here are the two that are the allergens in gluten. Gliadin is usually the one that's the most significant in terms of people who have actual allergies. But you can see this is one of the problems with wheat. There's a lot of actual gliadin and gluten, and that's where the allergies can kind of come into it. Whereas oats has a lot less. Maize has similar ones, but a little bit different. And rice has quite a bit, but again, they're different. So it's all of these that have some gluten in them. Sulfur is key. And for guys that are farming, the bakers want methionine, cysteine in bread, and you can see this is low sulfur, medium sulfur, and high sulfur. When bread is made, the gluten or the flour is mixed, and as it's mixed, the sulfur residues hook to each other and give you that stickiness in gluten. And it's the gluten that influences how big the air bubbles are going to be when the yeasts and so on produce carbon dioxide. So that influences wheat size. So cysteine and methionine are key. But more importantly, the plants in senescing puts a lot of sulfur, and when I show you the content of wheat, actually, a lot of sulfur in there, because methionine, which is the first amino acid that is produced in the biosynthesis of the sulfur amino acids, is cysteine, and then we get the other one, methionine and glutathione, which is key for stress management, are produced. The plant loads the seed with sulfur because it's key to germination in the spring. There are lipids in the germ, and that's important with wheat because lipids give you a lot more energy. And there's a whole bunch of stuff in the seed coat that is not so good for us. It's fiber for humans, but plants in the seed coat put in a lot of phytic acid or phytases, and that contains most of the P, but it also ties a whole lot of other cations which are not available to monogastric animals like cattle and chickens, and the cattle guys will know all about this, and the pig guys. So that's a bit of a problem, and I'll talk about that in a little while. Maturing seed can live a long time. We know that. It's stored a long time. And in the end, here's what we got. We got a pile of carbohydrate in the endosperm. We got a little bit of water. We got proteins galore. We got vitamins, minerals, and we got a tiny amount of lipid in the germ. And as you know, if you eat germ, that one of the things you have to be concerned about is rancidity of germ because of the fat in it, the levels in it. And we know that the seed takes up a pile of N and a pile of P and not so much K. So it's not surprising that we have a lot of that phosphorus and a lot of that nitrogen is moved into the seed. So the seed starts out with that. The same thing happens with soybeans. Look at all the protein, the phosphorus, going into the seed. A lot of the nitrogen going into the seed. So the plant moves that into the seed, not because the plant knows the farmer's going to use it. It does it because it wants the seed to use it. So the mother plant prepares the seed for a new life. So now we're into the seed to seedling. Here's what happened. Seed intakes water. Germination is completed when the radicle penetrates the seed coat. I'll show you that. Seedling dependent on stored reserve from anywhere from 7 to 14 days. And if you're planting canola at 2 degrees, It's going to keep that plant going for a lot longer, depending on how warm the soil is. There's sort of two embryonic kinds of elongations. There's stem elongations. There's a micropyle, and I'll show you that, in beans that allows water to come in. And some seeds have mucilage on them. Seed germination is regulated after imbibing water. First, it has to imbibe water, and that stimulates the messenger RNA to start producing hormones in this. I'll show you the pictures in the aileron and the scutellum of wheat. So gibberellins suppress ABA, abscisic acid. Abscisic acid levels rise in the fall and put the seed into dormancy. When we get imbibation, the genes are upregulated for gibberellins, and gibberellins suppress ABA, and we get breaking in dormancy. Then the plant starts remobilizing using enzymes and hormones are synthesized, as I said, in the scutellum and aileron, and then it starts breaking down the starches, the endosperm cell walls, the fats are broken down, and the phytases that contain this phosphate salts are released. Just the reverse of when it was putting in. And the proteins are broken down. Alpha amylase comes into place as an enzyme to break down that starch, and storage proteins are mobilized. Just that easy. Now, part of the education part, dicotyledonous plants don't exist anymore. If you're reading literature and you see eudicots, those are now, that's the new name for dicots. We still use monocots. That's just been a recent change. I'm not sure when that happened, but that's if you're reading things. So we're going to almost get to the pictures. I just point this out here. You can see all these plants are germinating, and why they all have some fats in them is because you get a whole lot more ATP. You get, 0.717 to, almost double the ATP out of fat compared to sugar. That's ATP from sugar, ATP from fat. So almost all of them will have them in their germ because they need a lot of energy to germinate. Epigeal and hypogeal plants, you don't have to remember that, but that's the name of them, that's all. That's the only reason, but I put this in here for one purpose. We have two plants that are epigeal, canola and soybeans. And you can see what that means is that the cotyledons are pushed above ground when they germinate. In the hypogeal types, the seed stays below ground. Here's P, and you can see the P. There's no seed up here. I'll show you more details. So the difference is that you can have a pea or a wheat plant freeze off, and as long as the growing point, which is down here, isn't killed, it'll regerminate. If you kill the growing point on canola up here, and I'll show you that, and the growing point right in here, in soybeans, plants are dead. So here are various plants. This is soybean, and you can see that the cotyledon is gone. Whereas with peas, chickpeas, lentils, the seed stays below ground. So here we go. On to the photos. We get germination and leaf development are the two stages I'm going to show you now. Here's germination. We get a dry seed, beginning of imbibation. Seed completely imbibed, the radicle emerges. The radicle elongates, root hairs and side roots are formed. The coleoptile emerges from the seed, and emergence occurs when the coleoptile penetrates the soil surface. So let's take a look at all that. Here's our seed. The seed imbibes about 45 to 50% of its weight in moisture. Genes are activated, as I said, hydrolytic enzymes break down stuff. We got oxygen in there and things are rolling.